Good morning again. So this is the second video dealing with reproductive traits, and it is about the flower morphology and function. I'm always Andrea Mazzucato from the University of Tuscia in Italy. As you know, the tomato flower is a typical hermaphrodite flower made up of 4 concentric whorls of organs: the sepals, petals, stamens, and carpels. The standard number of organs in the first 3 whorls is 5 or 6, whereas the innermost pistil is made up of 2 carpels. However, a large variation for the number of organs, the size, and the color exists among cultivated types. The first flower trait mentioned by the CPVO protocol is flower color. This refers to the color of the corolla and the staminal cone that are normally bright yellow. The protocol includes an alternative of the orange color. There are mutations of genes of the carotenoid pathway that cause an orange color of the flower, especially of the staminal cone. Sorry for this double flower, but I'm sure you can appreciate the orange phenotype. One of these genes is the lycopene beta cyclase that, when it is present with an active allele, makes both the staminal cone and the fruit to acquire orange color, as in the Orama cultivar mentioned by the protocol. Surprisingly, also a loss of function variant of the same gene, called old gold crimson, makes the stamen orange, although in this case the fruit is not affected. And this is the case of the pericherry cultivar mentioned by the CPVO protocol. Other mutations exist that vary the flower color in tomato. The tangerine mutation, for example, is giving an orange fruit and also produce an orange flower like the one we saw before. Yellow flesh, the mutation which is the basis for yellow tomatoes, has a very pale yellow flower. Green flesh, a mutation due to the effect in chlorophyll degradation, has a greenish flower. And finally, the white flower mutation, again affecting a carotenoid biosynthetic gene, has a whitish flower color. A trait which is taken into consideration by the CPVO protocol, and it is hidden within the flower, is the pubescence of the basal part of the style. To detect this trait, you have to dissect the anther cone and expose the pistil. The protocol gives 2 classes corresponding to absent or present. However, a wide variation of these traits naturally occurs. We actually never score this trait in our project because it's quite tricky to carry out in the field. A very important trait which is related to the flower peduncle is the presence or the absence of the abscission layer. This is trait number 19 in the CPVO protocol. There are mutants lacking this layer that are called jointless mutants, and these have been adopted in processing tomatoes because the absence of the abscission layer allows fruit detachment without the calyx and without the remaining peduncle segment, and these highly facilitate mechanical harvesting. Also in our screening we adopted 2 classes, although intermediate cases may be possible to genetic or environmental effects. Related to the abscission layer, and only for varieties with the abscission layer, trait number 20 in the CPVO protocol considers the length of the peduncle from the calyx to the layer. The protocol foresees 3 classes related to reference varieties. We actually never adopted or used this trait in our screenings. Some other important floral traits are not included in the official protocol, and one of these is the position of the stigma related to the tip of the staminal cone. Modern tomato varieties have the stigma well retracted inside the staminal cone. This ensures autogamy and fertility. However, many genotypes still show different degrees of stigma exertion that can range from the same level of the stamens, this is say the flush stigma, to slightly or strongly exerted. This is a qualitative scoring that can be done on several flowers and on trusses of different order per each plant. You can also detect this trait in a quantitative estimation by dissecting flowers as you see in the photos. Then single elements of the stamens, the filament and the anther, and of the pistil, ovary and style and stigma can be measured digitally or manually. And then stigma position can be expressed as the ratio or as the difference between stamen and pistil length. Additional flower variant phenotypes may be detected depending on known or unknown gene mutations. Frequent variants are those imposed by homeotic mutations that affect the identity of floral organs. Mutations in the so-called class B matchbox genes that encode for floral organ identity transcription factors cause the change of identity of petals into sepals and of stamens into carpels, as you see in the picture on the left. This gives rise to a male sterile individual. Other time, petals become sepaloid in a weaker way and for unknown reasons, as in this landrace in the photo in the middle. In the flower on the right, the sepal has lost its identity to revert to the feature of a leaf. Other mutations may affect the size or the number of floral organs. For example, the macrocalyx is a mutation enlarging sepal size. It can occur in cultivated material, such as in the landrace from Italy that you see on the left. The fasciation that consists in an increase in floral organs in all floral whorls can be already well visible on the flower, giving an increase in the number of sepal, petals, stamens, and even the carpels, as you see in the flower in the middle. Other variants may consist in aberrant stamens, or lack of proper deitions, and these cases gives rise to different types of male sterility. Finally, the ovary may undergo precocious development independently of fertilization, and so leading to seedless fruit. This is a phenomenon known as parthenocarpy. Then if you want to check the flower reproductive functions, you have to shift to the microscopic level. On the male side, staining for callose deposition and observation under a fluorescence microscope allows to check the progression of meiosis from the meiocyte, the cells on the left, to the formation of tetrads. To check the proper progress of microgametogenesis and pollen formation on mature flowers, pollen viability can be estimated with very simple stainings, for example with acetic uracil, and counting for normal and aberrant pollen grains. Finally, to check pollen germinability, you can adopt simple growth media to induce germination in pollen and to observe it with the callose staining again. On the female side, if you want to check the progression of meiosis, you can still stain for callose ovules after dissecting from the ovary. And you can see in the first two photos of the slide the tetrads formed at the end of meiosis. Then if you want to check the progression of gametogenesis and gamete formation, you have to go inside the tissue, and you have to rely on sections of flowers and microscopic observations. Okay, this is all for the reproductive traits. I wish to thank again the local organizer, the Maritza Vegetable Crop Research Institute, the Arneston Project and the Roxy Coast Action for supporting the course. And of course, I thank you for your attention and have fun with the flower phenotyping.